everyone. Welcome to our macromolecule video series. In this video, we're going to talk about nucleic acids. Nucleic acids are polymers of subunits called nucleotides, and they're used to store and transmit information. There are two main types of nucleic acid, DNA and RNA. DNA can direct its own replication and can also direct the synthesis of various types of RNA, including messenger or mRNA. mRNA, in turn, can direct protein synthesis, and this is the process that we call gene expression, getting from instructions in a gene to a functional molecule within the cell. All nucleotides have the same basic structure. They all contain a 5-carbon or pentose sugar, 1 to 3 phosphate groups, and a nitrogenous base. This just means that it contains a lot of nitrogen. There are a few different bases, which we'll look at in just a moment. The carbons in the sugar are numbered, with the 1' carbon being attached to the base and the 5' being attached to the phosphate. Even though they are both polynucleotides, there are a couple of important differences between DNA and RNA. First of all, they have a slight difference in the sugar that they use. In DNA, the sugar is deoxyribose, while in RNA it's ribose. If you look at the image here, you can see that the 2' carbon of the deoxyribose only has a hydrogen, while the 2' carbon of the ribose has a hydroxyl group attached. The second major difference in the structure of these two molecules is the bases, shown here on the top of the image. Both DNA and RNA contain 3 of the same bases: adenine, guanine, and cytosine, or A, G, and C. In DNA, the 4th base is thymine, or T, while in RNA, T is replaced with uracil, or U. The final structural difference is the number of strands. DNA is almost always double-stranded, while RNA is typically single-stranded. It's worth noting, however, that there are exceptions to these cases, and so it's better to use the sugar and bases for differentiating between DNA and RNA rather than the number of strands. The ways that the nucleotides are joined in a nucleic acid is by the formation of a phosphodiester bond between the sugar of one and the phosphate of the next to build what we call the sugar-phosphate backbone, illustrated by the blue ribbons on this diagram. The bases protrude out from the backbone, and in DNA, they can bond together to form structures like the rungs of a ladder. You'll remember we mentioned the numbering of the carbons around each sugar, from 1' attached to the base to 5' attached to the phosphate. When nucleotides are joined, they're always attached in the same orientation relative to each other, with the 3' hydroxyl being bonded to the phosphate of the next nucleotide. This means that nucleic acid strands have a direction, which we call 5' to 3'. When the strands are paired, they do so with one effectively upside down relative to the other. This is what we call anti-parallel, and it will be important when we come to look at the mechanism of DNA replication. Much of the function of the nucleic acids comes down to a specific property, which we call complementary base pairing. This means that each base has a specific partner that it bonds with, C with G, and A with T in DNA, or U in RNA. The reason for this specific pairing comes down to the functional groups on the different bases. C and G are able to form 3 hydrogen bonds between them, while A and T, or A and U, are only able to form 2. Thanks for watching, and please be sure to check out the other videos available in this section.